The nervous system. The nervous system is divided into two parts, the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and spinal cord, where information analysis occurs. The peripheral nervous system mostly consists of nerves, such as sensory and motor. Your nervous system is in charge of triggering involuntary responses from your body acting on information, like answering "what's your name" or 2 + 2 = 4. The nervous system consists of interconnected nerve cells called neurons and glia. Their job is to generate electrical signals called action potentials, which allow them to quickly transmit information in the body. A neuron's cell body is called a soma, where the nucleus is also found. It consists of dendrites, which are just extensions of a neuron that carry electrical signals from synapses to the cell body. Axons are nerve fibers that are capable of rapidly conducting impulses away from the cell body. Within the axon is the process of action potential. Lastly are axon terminals, which are located at the end of the axon. They contain neurotransmitters. The nervous system contains 3 types of neurons. Sensory neurons carry signals from our sense organs to the central nervous system. They are also known as efferent neurons. Interneurons connect various neurons within the brain and spinal cord. Motor neurons carry signals from the central nervous system to the outer parts, which are muscles, skin, or glands of our bodies. They are also known as efferent neurons. Within a sensory neuron's dendrites, an impulse is passed through its axon, making the axon rise from its axon hillock. This is known as the start of action potential. Ions are chemicals in the body that have an electrical charge. They are located in the nervous system. The significant ones in this case are sodium and potassium because they play a role in action potential. On the membrane of the neuron, you will find ion channels that are closed, which tell you that the cells are at the resting potential. The only way the neurons are able to create an action potential is when the concentration difference of ions between the intracellular space and the extracellular space are in its resting potential. Threshold potential triggers action potential, which is when there is a higher concentration of sodium ions outside of the neuron and a higher concentration of potassium ions inside of the neuron. The extracellular space is more positive than inside the neuron because of the sodium and potassium uptake pumps that regulate the environment by pumping out 3 sodium in exchange for 2 potassium ions. When a neuron is stimulated, it causes sodium channels to open, letting positive ions in. This changes the electrical environment to make it more positive inside the neurons and less positive outside. This causes depolarization, which causes a chain reaction where the next sodium channels will open, letting in positive ions all the way down the axon. Shortly after the channels have opened, they will close again, and potassium channels will then open, letting out positive potassium ions to recreate the negative environment inside the neuron and the positive environment outside. This causes repolarization, where the intracellular space becomes negative again. The neuron then reaches a state of hyperpolarization, where the cell has let out too many ions and has now become more negative than the cell's resting potential. This is also known as the refractory period, because the threshold stimulus will not trigger another impulse on an axon, its recovery phase. But this is quickly corrected by the sodium-potassium pump, which then the neuron stabilizes at its resting potential. In order to transmit a signal between 2 neurons, an electrical impulse must be communicated over a synapse. When action potential begins in a neuron, it travels down an axon. When action potential reaches the axon terminal, calcium channels open and calcium ions rush into the neuron, causing the neuron to make and store neurotransmitters in the vesicles. When the vesicles contact the axon terminal membrane, the neurotransmitter is released into the synaptic cleft. This causes the postsynaptic neuron receptors to activate. In this case, these receptors allow sodium in the neuron by facilitated diffusion, causing an action potential to start in the postsynaptic membrane. The neurotransmitters are released from the receptors and diffuse back into the synaptic cleft. The vesicles then recycle some neurotransmitters to prepare the neuron for its next action potential. When the sensory neuron has finished its synaptic transmission, meaning that the impulse is now in the inner neuron, it is now the inner neuron's turn to go through the same process as the sensory neuron. This synaptic transmission can go one of two ways, either through a divergent or convergent pathway. A divergent pathway is when one presynaptic neuron branches to affect a large number of postsynaptic neurons, usually during the synaptic transmission of a sensory neuron to an inner neuron. A convergent pathway is where many presynaptic neurons converge to influence a smaller number of postsynaptic neurons, usually during the synaptic transmission of an inner neuron to a motor neuron. The difference between the processes of all three types of neurons is that the inner neuron dictates whether the motor neuron will be excitatory or inhibitory. Excitatory meaning that neurons will generate the impulse to the effector, or inhibitory meaning that it will not carry through. The sum of excitatory and inhibitory signals dictate the neuron if it's going to fire the impulse or not. If the neuron does fire, it is known as excitatory postsynaptic potential, but if it does not, it is inhibitory postsynaptic potential. This process happens during synaptic transmission from the inner neuron to the motor neuron. If the neuron decides to be excitatory, again meaning that it is going to fire the impulse, it is now the motor neuron's turn to go through the process of action potential, then through synaptic transmission before it can bring the impulse to an effector, either an organ, gland, or muscle. This entire process is known as a reflex arc. It is our brain's way of letting our bodies react to outside and inside information.